Thanks, Susan. All right. Well, thanks everybody for joining us this morning for the first inaugural My Field for Dummies talk. It's my pleasure to be here with you today to tell you a little bit about the field of microbiome research. And of course, I work on bats as a person who is affiliated with the GBATnet consortium, but the things that we're going to talk about today are actually broadly applicable to any wildlife system that you might want to study. So hopefully you'll enjoy the talk. So I just want to get started by kind of giving you a background to how I came to this research and what kind of background I have. So as Susan said, I'm currently an assistant professor at Fairleigh Dickinson University, and the kind of research that I do is highly integrative because it unites the fields of microbiology and kind of bench science, molecular biology, with fieldwork in natural history research. So I have 2 feet in very different worlds here. I'm a field biologist, but I'm also a bench scientist as well. So I've been working with bat microbes virtually for my entire career. I did a master's at Fordham University where I was studying white nose syndrome, which is caused by the fungus Pseudogymnoascus destructans, which of course causes the deadly white nose syndrome disease in North American bats. So I spent a long time studying one particular fungus and the disease that it causes in bats. And it made me curious about the other microbes that call bats home and what they're doing, not necessarily just causing disease, but how they might actually be supporting bat health. So that's why I did my PhD at the American Museum of Natural History, trying to get a broad survey of the bat gut microbiome across different ecological feeding niches. So what are the resident gut bacteria that live in these animals? And then later, for my postdoc, trying to develop this idea of what are those microbes doing functionally on behalf of their hosts, which is still a research program that I'm pursuing today. And as I mentioned, I'm not just a bat person when it comes to this research. You can apply the techniques that we're going to talk about in today's webinar to many different wildlife systems. So some collaborative projects that I've used these same techniques on are studying the effects of nest parasitism on the microbiota of baby tree swallows and bluebirds in collaboration with Sarah Canuti at University of Connecticut, as well as using very similar techniques in environmental DNA research to monitor the restoration of oyster beds in New York City estuaries with NYU biologist, the team of NYU biologists, basically. So I'm going to break this webinar into 3 parts broadly. So first, I really want to start at a very basic level by giving you guys an introduction to the microbiome. What is this concept? What are we talking about when we're studying these types of things? I'm also going to give you kind of a how-to guide for how you might approach this kind of research in bats. So this is not a completely exhaustive webinar. There's lots of choices that you'll have to make along the way. But this is my attempt to give you a tutorial and a starting place if you're interested in doing this kind of research yourself. And then at the end, we will talk about how you can apply these microbiome concepts to the study of ecology and evolution in bats, but also in any other kind of animal you may be interested in, which I hope will spark some good discussion questions from you all at the end. So let's get started with part 1, dissecting and diving into this concept of the microbiome. So I want to start really, really basic here because I understand that not as many people are thinking about microbes as often as I do. So when we're talking about the microbiome, microbiologists may be interested in studying lots of different domains and kingdoms of life under this umbrella. So we may be interested in studying, oops, I don't know why these are auto-advancing. We may be interested in studying bacteria and archaea, which is currently what I mostly work on. In the past, I've also studied fungi, as I mentioned, although unicellular protists also count as microbes, and they may be included in this microbiome concept. And then, of course, we may be interested in studying viruses or virions. So these are infectious particles that may have a DNA or RNA genome, but are not usually considered living things, and yet they still fall under this microbiology umbrella, and therefore could be studied in these microbiome research projects. So these are the taxa that we're interested in. And you may be familiar with the fact that this is kind of our best hypothesis coming from Carl Woese's paper back in 1990 of how these organisms are related to each other. So if you're not used to looking at a phylogenetic tree, the way that you read this thing is basically from the root node here to the tips. So we have past ancestors here, closer to the roots of the tree, and the tips are getting us more towards present species, okay? So for the most part, bacteria are going to be our most ancient lineage of living things. And I think this is a really important concept because it implies that all animal life evolved in a microbial world. And it's because of this that most animals are colonized by a vast array of bacteria. Oh, and then of course, anywhere you see these things joining together at a node, this implies it was a last common ancestor. So here we see that archaea and eukarya are more closely related to each other than they are to bacteria because they share a more recent common ancestor. So you may see some trees in this talk. I just want to make sure everybody's familiar with how to read them. So before we actually define this concept of the microbiome, I wanted to give you a really brief history lesson, which was that ever since the very first microbes were discovered by Antony von Leeuwenhoek, looking in his microscope at his own tooth plaque, our relationship to bacteria was mostly defined by their capacity to cause disease in humans. So when I teach microbiology to my students here at FDU, I always give them this discussion of Robert Koch, who was the biologist that came up with the disease diagnostic criteria known as Koch's postulates, and discovered that Bacillus anthracis was the causative agent of anthrax disease. So, you know, for centuries, this was our relationship to bacteria. We're thinking about them as individual species that are capable of causing disease phenotypes in humans. But over the past, I would say, 3 decades or so, we have now developed this understanding that human and animal-associated bacterial communities, which are known as the microbiome, are inseparable from who we are. So it's not just one species causing one disease anymore. We now have this kind of community ecology concept applied to microbial communities that live in and on us. And we recognize that these communities are not only commensal, they're not just along for the ride, but most of the time they're actually doing things on behalf of our bodies that we could not do ourselves. So I may also use the term symbiosis when talking about these communities, so I just wanted to define that term as well. This is just 2 or more organisms living in close association with one another. The relationship doesn't have to be parasitic or mutualistic. As long as they're living in close association, it counts as a symbiosis. So what are some of the things that the microbiome can do on behalf of the host? They're pretty essential in food metabolism. So many of the short chain fatty acids that your body produces on a daily basis or consumes on a daily basis were actually generated by intestinal microbes living in your gut, not by your body's own metabolic activities. The gut bacteria also help regulate and promote immune function. So bacteria in the gut live in close association to these toll-like receptors, which are direct links to the host immune system and help to stimulate the proliferation and differentiation of immune cells throughout the body. And there's also increasing evidence that our gut microbes can alter and influence our behavior. So 95% of the serotonin neurotransmitters that are produced in your body on a daily basis come from the activity and metabolism of microbes. Bet you didn't know that. So these communities are really complex, and they are so tightly integrated with the host that there are all these potential feedbacks between the host and the microbiome that even in humans, we are only really just starting to fully understand. In terms of animals, we have a lot of evidence of this kind of symbiosis and mutualism in lots of different species, but most of them are the ones that are relevant for humans, right? So agricultural and pet animals. So one of the best studied microbiomes on the planet is probably the cow-rumen microbiome. Cows, as we know, eat grass, which is high in cellulose, which is an undigestible polysaccharide for mammals. And so they actually rely on the chemistry and the metabolism of bacteria and archaea in their multi-chambered stomachs to break down those cellulose cell walls and make them into carbohydrates that the cow can actually use. But when it comes to the microbiomes of wildlife that are not agricultural or commensal species, we really have much less of an idea of what is going on with their microbiomes and how they may or may not rely on them to accomplish similar metabolic or immune-related goals. So because I'm a bat biologist, of course, this was the system in which I wanted to explore these kind of questions. And so what I'm going to do today is kind of give you a roadmap to how you can ask similar or perhaps even more novel questions in the bats or other wildlife animals that you would want to study. So that will move us into part 2, which is discussing this how-to guide for microbiome research in bats. But like I said, almost all of these techniques can be broadly applicable to any animal system that you may want to use them in. So the reason why I chose to study the microbiomes of bats is because since we know that the microbiota is really inextricably linked to the metabolism and nutrition of the hosts, I thought this would be a really interesting question to explore in bats because they are so ecologically diverse. As many of us already know, bats have radiated into every terrestrial feeding niche that it is possible to fill as a mammal. And so you can really sample them across all possible dietary guilds to ask these kind of questions. So the hypothesis that my research mostly focuses on is how bats are able to specialize on these nutritionally difficult food resources. So particularly things like fruit, blood, nectar, etc. So the hypothesis that I explore in my research is that, bats eat these food items which may not be nutritionally complete. So suppose you have this little sternira eating a fruit. The bat is going to use the nutrients from that fruit for its own digestive processes. But those same nutrients could also serve as substrates for the metabolism of the microbes that live in their gut. So microbes can do a lot more metabolic pathways than mammals can. And so they can potentially liberate metabolites that the host would not be able to produce on its own. But because they live in this close interface with the gut epithelium, the host can actually absorb some of those nutrients produced by their microbes, therefore perhaps supplementing their nutrition from just the food items they ate alone. So this brings us to the question, okay, we have an interesting research question like this. How do we actually go about doing this kind of study? What does it entail? So in this roadmap that I'm about to share with you, I've kind of broken things down into 3 kind of big steps in this pipeline. The first is specimen collection, right? So we need to go out and get samples of the gut microbiomes of these animals. And then we need to get a sense of who's there. So we're usually going to do this using genetic sequencing techniques because many microbes are impossible to grow in the lab. Something like greater than 70% of all microbes on Earth are not easily cultivable. So we're going to use genetic techniques to profile them instead. So we'll talk a little bit about genetic library preparation and sequencing and some of the options that you have there, depending on what your research question is. And then we'll briefly talk about data analysis. So once you get all these gene sequences back from your sequencing facility, what do you do? How do you turn those raw genetic sequences into data that we can actually answer these questions using? So let's talk about specimen collection first. So when you go out to the field to collect microbiome samples from wild animals, you basically have 2 options. You can either collect feces from the animal, which is most of the time, this is what I do, or intestinal contents, depending on your other project goals. So I wanted to kind of give you a little bit of a sense of the pros and cons of both of these approaches, because they're very similar in terms of the data you get at the end, but they do have some limitations. So the pro of collecting a fecal sample is that it's really easy and it's non-invasive, right? So you can put a bat in a bat bag and wait for it to defecate and very quickly shuffle that poop sample into a tube with preservative. So it's very quick, very easy, and does not stress the animal out very much. And if you think that you have a high probability of recapturing animals from one field season to the next, this approach allows you to do repeat sampling. So if those animals are pit tagged, as we are beginning to do in Belize, where I work, this means that you can actually sample these individuals through time over their adult life. And then, of course, if you choose to collect intestinal contents, the main pros of this approach is that if you're interested in doing microscopy to like examine the spatial arrangement of where bacteria occur in the gut lumen, you can do that, right? Because you have the tissue preserved. This method is also kind of nice because you can assess other what I sometimes call macro microbes, like the eggs of worms and things like that. So if you want to look at parasites and you have the entire length of the gut, you can do that too. So you get a little more bang for your buck, perhaps, with intestinal contents. But of course, there are some cons to these approaches. So one of the cons of collecting a fecal sample is that depending on how quickly you can get to it, these samples can be kind of more prone to environmental contamination. So even inside of a bat bag, there's lots of DNA floating in the air. Beth Claire has done a lot of great work showing that actually there's quite a lot of DNA from quite a lot of species that you can detect in the air. So it's very easy to get some kind of airborne contamination on a fecal sample. The cons of intestinal contents are a little bit more obvious, I think. So this is a lethal sampling method. You have to take the intestines out of the animal and it needs to be dead for that to happen. So this is not a suitable approach for working with sensitive or endangered species. And of course, if you're sacrificing the animal, you can't sample the same individual over time because you get one shot, right? You've sacrificed the animal, now it's dead. But of course, choosing one of these two methods is going to depend on your sampling goals, the kind of research questions you have, and the larger context of the research that you're doing. So if you're a museum person like I am, and you're collecting voucher specimens for the museum, collecting intestinal contents would make sense if you're already sacrificing the animals for taxonomic vouchering. So one of the questions I asked really early on in my research career is, well, are these communities the same? Because for the most part, I would prefer not to have to sacrifice animals if I don't need to. So I actually did a study on this and found that they're pretty similar, these two sample types, although fecal samples tend to be a little bit more diverse. I think the reason for that is because if you kind of look at this little drawing I did of the spatial arrangement of bacteria in the gut, you basically have two subsets of that community. So you have the bacteria in the lumen, which is kind of the empty space inside the tube of the intestine. And in this community or subcommunity, you may have bacteria that were ingested along with the food items. This is where enzymatic digestion is happening. So you may have very particular microbes occurring here. And this is the bulk of what's going to get excreted as feces, okay? But contrast that with the endogenous intestinal bacteria that are actually affixed to the mucosa of that gut epithelium. These species are the ones that are going to interface more directly with the immune system because the epithelial cells and all those toll-like receptors are located right here. And they're also going to tend to be adherent to the mucosa, meaning that unless you agitate this area with a swab, for example, only a very small proportion of these bacteria are actually going to be excreted along with the feces. So there are differences here, but not enough to kind of swamp out any ecological or phylogenetic signal that you may be hoping to detect. Okay, so that's the first fork in the road, right? What do I sample? So once you've made your choice, whether you're going to sample feces or intestinal contents or swabs, it doesn't really matter. The next choice you're going to have to make is, how do I preserve these communities? Preserving microbiomes is a little bit different than preserving tissues for genetic work from the host, because microbial communities can continue to actively replicate after sample collection is complete. Meanwhile, if you take a liver sample, you're not going to have any real, you can have degradation, but you're not going to have changes to the structure of the cells in the host liver. So what we want to do is, once we've collected our microbiome sample, we need to make sure that A, we are halting all bacterial chemistry and reproduction, and preserving the nucleic acids that we'll need to build our genetic libraries later on. So I have given you kind of a range of options here that people employ when they do field research. And of course, coming from the human microbiome literature, we now know that liquid nitrogen with no other preservative is kind of the gold standard in this research. So if you take a microbiome sample, put it in a dry tube with no other preservative, and plunge it directly into liquid nitrogen, you're not only preserving the DNA and RNA in that sample, but you're also ensuring that at these super cold temperatures, you are halting any chance of specific bacterial taxa continuing to survive and reproduce. Of course, this is not always a feasible preservation method for those of us who work in remote field locations, right? So we have to have some other options. One of the middle of the road options that I think is a really good choice is this DNA RNA shield preservative, which is made by Zymo Research. This preservative is pretty economical. You can get about a 200 milliliter bottle for under $300 US. So it goes a long way. And it does two very important things. First, it halts the reproduction of microbes just like liquid nitrogen, and also renders them biologically inert. So you can get, a disease agency permit, like in the US, we have to get a CDC permit to import bat samples, and they want to make sure that the stuff is not infectious any longer. So DNA RNA shield achieves both of those things. It also offers some ambient temperature stability, so you can keep your samples at room temperature for up to 2 weeks in DNA RNA shield, and then put it in the refrigerator. So we're talking about 4 degrees Celsius here, and it can stay in there for months and still protect the DNA and RNA, which is a much more feasible option for those of us working in really remote locations. If you're in a super remote location where you may not even be able to access a refrigerator, another option is to use Whatman FTA cards. So they're little booklets that look like this. They're a little bit more on the pricey side, about $500 for a pack of them, but they have multiple little circles where fecal material can be preserved and kept dry. So basically you just put this in a little sandwich bag with desiccant, and it can stay at room temperature indefinitely until you're ready to extract that DNA. So this is a great option if you're working in a really remote location, you're going to be out in the field for months and have no access to any kind of cold chain at all. FTA cards are great. 95% ethanol is something that people used a lot during the early days of microbiome research, but we now know that 95% ethanol is not a great preservative, because proteobacteria, which are a huge phylum of bacteria that are very common in bats, can survive and reproduce even in 95% ethanol. So you get what are called proteobacterial blooms, where you have proteobacteria taking over the sample, and that is not actually reflective of the host biology at all. It's just an artifact of how it was preserved. So I never recommend 95% ethanol unless it is the only option available to you due to constraints of finances or being able to get any other kind of preservative. So once you have your samples nicely preserved, transported back to your lab, now you have to get the DNA out of the microbes, right? So in general, there's lots of commercially available kits that can achieve this. But what I recommend is that you just make sure that no matter what kit you use, it should have both a chemical and mechanical lysis step. So most regular tissue extraction kits do not have this bead beating step, this mechanical lysis, but you need to have that because bacterial cell walls are really tough and difficult to break open. So both of these two kits, these are just some examples, both of which I've used before. the QIAGEN DNeasy Power Soil Pro Kit, and the slightly cheaper Zymo-Biomics DNA Mini Prep Kit. Both very similar chemistries. They both give pretty much interchangeable results. The other thing these kits have, which is not typical of other extraction kits, like just the regular DNeasy kit, is that these kits have this humic acid precipitation step. So as you can see in this picture here, these are two fecal samples, one from a fruit-eating bat, one from an insectivore. And they have a lot of stuff, you know, the brown kind of humic acid stuff or plant secondary metabolites that have been excreted along with the feces. These things will inhibit your PCR. And so if you don't use a kit that has this humic acid precipitation step, the DNA will not be suitable for PCR amplification. It may look clean when it comes off the column, but the presence of those humic acids in the fecal samples will interfere with your ability to amplify whatever gene you're targeting. So let's say you use one of these kits and you have perfectly clean DNA free of any humic acids. The next choice you'll need to make is what kind of sequencing technology you want to do to profile these microbiome communities. Remember that we're dealing with a mixed microbial community. We have already achieved our DNA extraction. And now the question is, will we go with amplicon sequencing? So this is often known as 16S sequencing. Or will we do metagenomics sequencing and sequence all of the short sequence fragments from any DNA that was present in that sample? So this is a choice that you will ultimately have to make, and it will largely depend on the budget that you have to do this project, how much is known about the microbial communities you're interested in, and the specific questions you have for your research. Amplicon sequencing, or 16S, is a very simple approach, and if that microbiome community that you're working with has never been profiled before, so if this is a species for which we have none of this kind of data, this is a really tractable place to start. So once you have your DNA extracted from those samples, to do the 16S library prep, all you do is basically amplify this one gene, 16S, which serves as a barcode that can be compared to a database to identify all of the bacteria found in that sample. So I won't go into the details of those library preps, but I have tons of protocols I can share with anybody who'd be interested in reading them. But once you have those 16S libraries constructed, so for each sample, you're going to have a bunch of these 16S amplicons, which came from a variety of bacteria that were present. Then you'll mix these libraries together, normalize and pool them for sequencing, and finally sequence, which can be usually done on a Illumina MiSeq platform. And so at the back end of this, you'll get these giant, multi-gigabyte FASTQ files, which are just strings of A, C, T, and G, right? And then what you'll need to do is apply bioinformatics. So there's lots of really standard pipelines out there, which we'll discuss a little bit more in a moment, that can be used to separate these sequences back into the samples they came from, filter them for quality, throw out any reads that were garbage, and then analyze the data to ask kind of more hypothesis-driven questions about them. So the program that I use to do this mostly is QIIME2, which we'll talk about in just a minute. So QIIME2 I really like because it's an open source and free to use pipeline, and the developers maintain an incredibly active forum so that if you ever run into issues trying to analyze your data, you can talk to the developers and they'll get back to you very quickly. They're great. The other 16S pipeline many people use is called Mother. I'm not as familiar with this one because I haven't used it myself, but it is fairly common and also open source. So the other sequencing option you have here is shotgun sequencing. So shotgun sequencing is different than amplicon because in the shotgun pipeline, instead of amplifying a particular sequence and then trying to check against a database to see what they came from, you're going to just sequence all of the genes present in that sample and then try to assemble the genomes from which they came at the end. So this process starts the same. You're going to extract your DNA and clean it up, figure out how much you have. Where it's different than 16S is that you're going to, instead of doing this PCR amplification, typically you're going to end up shearing the DNA to a certain size using a machine like a Covaris. You may have a couple of cycles of PCR to put on these adapters, which you'll need for sequencing. And then very, very few, so only 15 cycles of PCR amplification to yield enough DNA to actually put on the sequencing machine. And there are some shotgun workflows that are actually PCR free. And then you can sequence it on something like a MinION or a HiSeq, whatever kind of library prep protocol you are using. Now, shotgun sequencing is a little bit more complex because, like I said, you have to reassemble all of these genomes that you've collected all these little fragments from, figure out who they came from, and then build your taxonomy that way. So there's far more assembly and annotation pipelines for shotgun than there are for 16S. And I've given you just a smattering of them here. They have different strengths and weaknesses, which we probably won't have time to talk about today, but these are probably the most common ones that I'm familiar with. Many of them achieve similar, if not the same, results. So I don't want to get too in the weeds here, but how this kind of works is that you have these contigs, these segments of DNA, and you're going to basically try to align them to each other to build what are known as K-mers. So they're going to try to find these little fragments and align them to each other to build the longest contig that you can. So once you have those contigs constructed from all these K-mers, basically, you can compare those to a reference database and try to figure out which bacterial species they came from. So because it can be difficult to decide which of these techniques is right for you, this is a flowchart I made for a metagenomics course I taught at the American Museum of Natural History, which I hope will simplify the process. This is at least the logical workflow that I use approaching this kind of work. So the first thing to ask yourself is anything known about the gut microbiomes of the species that you're working with? If the answer is no, just do 16S. It is cheaper. For reference, my 16S libraries, to have a facility prep the libraries and sequence them cost me about $19 US per sample. Shotgun metagenomics costs about $149 US per sample, but it is getting cheaper all the time. So if nothing at all is known about your community of choice, just do 16S as a starting place. If something is known, and there's a good reference genomic database for those bacteria, you might be able to do shotgun. But if the answer is no, then you should still probably do 16S. If something is known about your community, there's a decent database, and your research question is mostly taxonomic, you still might want to do 16S. But if you want to get after function, that is really the place where shotgun metagenomics shines. So as you begin to build your microbiome research career or project, you may find yourself using 16S and shotgun, but at different stages in that project's development. So I am at the point where I'm pretty much ready to only be using shotgun metagenomics, but it's taken me 5 years to get to that point, and many studies to build that kind of foundation of data to be able to ask functional questions. So a couple things I just want to point out about shotgun metagenomics is that you really have to consider some things that make it different than 16S. So 16S protocols are really standard. You can get them from the Earth Microbiome Project. Illumina publishes 16S workflows. There's not a lot of variability or decision points, I guess I would say, in 16S. With shotgun, it's a little bit different because it's really hard to estimate how many reads you need, so how much sequencing to buy. If it's a host-associated sample, which in my case it almost always is, a lot of those reads are going to be lost to sequencing host DNA because bats shed their own DNA in their feces as well. And this can be a huge proportion of the reads that you get out of a shotgun analysis. So if you wanted to do this kind of work and perform a de novo assembly, the best thing you can do is just sequence a sample on a paired end Illumina high-seq lane and try to get as many reads as you can. You also have to think about read length. So for de novo assembly, you want the longest reads you can. So 1 by 150 or 2 by 150 would be great. But if you're only interested in measuring how much of each bacteria is there, 1 by 90 or 1 by 100 is fine. So with shotgun, it's more expensive. There's a lot more decisions that you need to make. So I always recommend people start out with 16S and incorporate shotgun when they have some data to build on. So I'm not going to get too in the weeds, but what you need to know about shotgun analysis is that it has 3 steps. Quality control is the same as 16S. You want to throw out any junk reads. Where it's different is this assembly. So remember, we're taking really short reads and trying to build these long contigs using K-mers or other types of analysis. And then you're going to organize those sequences into bins, which will eventually represent one organism, we hope. So we're separating out these contigs and trying to figure out which ones belong with which so that we can assign those a bacterial species later. So I have a whole bunch of quality control programs here. You can see that choices are endless, and I'm not going to get into these, but I just want you to be aware of them so that if you're totally lost and you don't know where to start, you have a list of things you can start exploring. And the reason why shotgun is really good for asking these functional questions is that once we've done the assembly and the binning, now we have this genomic catalog, right? So each bin represents one species. We can take all the genes that we found in each of those bins and ask questions about beta diversity. So how do microbial species change from one sample to the next? We can use functional orthology to try to understand what the functions of those genes are, what pathways they take part in, and ask questions like, how are the microbes in native forests different metabolically than those in agricultural soils? And if you have a really robust data set, you can start trying to hypothesize these syntrophic interactions. So metabolites from one bacterial species feeding the metabolic properties of another. So these are the kind of questions you can really get at with metagenomics. But there are some challenges to this approach. So uneven abundances mean that you'll get highly non-uniform reads coverage across different genomes. And if you have microbial species that have a lot of repetitive DNA sequences, which is really common in certain microbial taxa, you can have problems correctly placing them. We also have to mention that there's a lot of issues with even defining what a microbial species is. Don't ask me because I don't know. And most microbial genomic databases are heavily biased to humans and other medically relevant bacteria. So if you're working in a natural history kind of context, working on bats and what their bacteria are doing, you may not get great matches to these genomic databases. Although that may not be the case in 10 years as things become more and more complete. In any of these studies, I would recommend considering your computational power. So a standard laptop lacks the CPU power to assemble even small metagenomes, but you can do small 16S studies on your local machine. Raw data files are going to be really large. And as I mentioned, there are streamlined pipelines that are less computationally intensive, but you will still require a high-powered computing cluster access to complete shotgun analysis. Okay, so that leaves us a few minutes for part 3, which is pretty short. I just wanted to share some applications of this research, especially as it relates to ecology and evolutionary biology, because I'd really like to hear from you all in a few minutes. to see if you have any questions or want to discuss ways that you think you could incorporate these techniques into your own research. So one of the really cool questions that we can ask is, how do we use microbiomes to understand host ecology? And I'd like to shout out my colleague, Kelly Spear, who did a really, really cool study looking at how changes in the quality of habitat can actually cascade through multiple scales of biological organization. So we're affecting the bats that live in these habitat fragments, which is affecting the parasitic bat flies that live on them. And those changes in that broader habitat can actually even be detected at the level of the microbiomes of those parasites. So we have this nested system, and at each level, at each scale, we can see how the organisms that are present there are responding to the loss of their habitat. So you should definitely read this paper. It's a really cool one. And this has direct implications for diseases vectored by these blood-feeding arthropods. So bat flies pass bacterial species like Bartonella among bat hosts, or at least that's what we think. And so if their microbiomes are changing in response to the broader things that are going on in the habitat, how does that affect the health of the bats that are harboring these parasites? We don't know. And this is a really good question to be asking, especially in the context of COVID-19. We can also ask cool questions about how microbes facilitate animal dietary ecology and how they may have served as novel sources of adaptive potential over time. So fishing bats like Noctilio leperinus and certain species of Old World Myotis actually have gut microbes that help break down omega fatty acids that are found at high abundance in fish, but not in insects. So this is a really cool study that was published in Animal Microbiome a couple years ago. And the findings were that these fishing bats have bacteria that are actually facilitating their dietary lifestyle that were not acquired from the environment, but are actually passed vertically from parent to offspring. And so this has direct implications for host evolution and adaptation, because it implies that microbes may be facilitating bats' ability to radiate into these novel ecological niches over evolutionary time. Okay, and this is like a brand new paper. It literally just came out this month, and I thought it was really cool, so I had to put it in here. So how can we understand host immunity through the lens of the microbiome? Really cool paper in Transboundary and Emerging Diseases, which found that if you inoculate germ-free mice with the microbiome of Hippocyterus armiger, those mice actually have lower infection with H1N1 flu. And the presence of the bat microbiome in those mice actually helps to lower the amount of lung damage they receive from infection with H1N1. These authors also did metabolomics to show that the bat inoculated mice produce more flavonoids and isoflavones than the mice which were either colonized with the normal mouse microbiome or remained germ-free. So this implies that something going on in the Hippocyterus armiger bat gut microbiome is actually regulating immunity, definitely in these mice, but possibly also in Hippocyterus, via cytokine secretion, because flavonoids and isoflavones are directly related to this upregulation of cytokines. So really, really cool applied questions we can ask using microbiome research. And to finish up, I just really want to plug that microbiome research in wildlife is a growing field. And I think I can speak for the entirety of the field when I say you should come and join us. If you want to do that in a bat-specific way, I, along with a couple of other colleagues, Elise Lauterbur, Hernani Oliveira, Mudasar, and Adil, are actually all co-group leaders in the Bats in Habitats, Bats has Habitats working group, which is a working group facilitated by GBatNet. Thank you very much. We meet virtually and all are welcome. We're always looking for new participants or group leaders if you have the time and desire to do that. So please email me, because I'd be happy to get you on the mailing list if you'd like to join us. And with that, I would love to turn this over to the group for questions and discussions, and I will take down my slides so that we can see each other instead of my slides. Thank you.
Hey, thank you very much, Melissa. Round of applause from everybody from the crowd. If there are any questions, feel free to raise your hand or type it in the chat and we will get to that. Are there any questions from the crowd? Melissa, why don't you explain a little bit about some of the objectives of the working group that you are co-leading?
Sure. So the Bats in Habitats, Bats as Habitats working group is trying to really flesh out this idea. So remember the figure I pulled from Kelly's paper, which shows that there are these kind of multiple scales of biological organization, and bats are just one level of that, right? So bats live in broader habitats, but they also serve as habitats for ectoparasites, microbes, and lots of other things, right? So the objectives of this working group is to kind of publish a review paper to explain and flesh out this phenomenon. So try to, organize these ideas of what does it mean to have these multiple scales of biological organization, with the hope that we can use this paper to apply for federal or international grant funding to study this multi-scale system in one or two really common and widespread bat species. So something like Rosetus aegypticus or something. So that's our 1 and 3 year goals for our working group. And we're making really good progress towards organizing these ideas for the review paper. So we'd love to have, more input from people with different backgrounds and access to different samples who may be interested in joining.
Right, and as a reminder to everybody, you can always join by signing up on the GBATnet website or emailing the group leaders, and they will be able to add you to the members list also. We have a question in the crowd from Joanna. 2 questions. First question, why only investigate bacteria and not also other microorganisms? Second question, is there a positive correlation between alpha diversity and functional role of the microbiome, meaning more diverse equals microbiome enhances bat nutrition more?
Those are really great questions, Joanna. So to answer your first question, why only investigate bacteria? Numerically, bacteria are the most abundant members of the gut microbiome. So archaea make up a much smaller percent, like less than 5%. Protists and fungi make up an even smaller percent, probably less than 1%. So numerically speaking, the kingdom Eubacteria is the major workhorse of the gut microbiome. But other body habitats, that is not always the case. So of course, there are more fungi on the skin. And it's totally possible to assess those communities as well. You just have to use a different marker. So everything I shared in terms of how to build those libraries, sequence them, analyze them is the same. But for fungi, for example, you would just use ITS instead of 16S. To answer your second question, is there a positive correlation between alpha diversity and functional role? That is a good question that I am not 100% sold one way or the other on, but my intuition says probably not, only because bacteria are characterized by a high level of functional redundancy, meaning that you can have a very diverse community of microbes that are doing mostly the same thing. So very taxonomically distinct, non-closely related bacteria can have the same metabolic pathways, which is slightly different than macroecological communities, right? There's not as much functional redundancy there. So it's possible that more diverse communities have more functions, but it doesn't necessarily need to be that way. Really good question.
Are there any other questions? Nobody from the crowd?
I've got one, Susan.
Go ahead.
Great talk, Melissa. I was wondering, I was at a talk last week for somebody who studies amphibians, and he was mentioning how the microbiomes have been associated with perhaps protection against chytrid fungus for certain microbiota. I was just wondering, do you know of any kind of associations like that in the bat world that you would mind sharing?
Thanks, Teresa. That's a really cool question. And it turns out that this has been studied in bats, particularly with white nose syndrome. So there are bacterial taxa that seem to be able to confer at least some antifungal properties to the bats that host them. Virginie Lemieux-Lebonte, who's up in Canada, did her whole PhD on this, and it's some really cool stuff. So you should look at her work. But if I'm not mistaken, one of the genera of bacteria that has these antifungal properties is Pseudomonas, which is a really common species. We work with it here in the micro lab for my students. And there's another one I think is called Rhodococcus. And so there's been some attempts to see if we could inoculate North American bats with these bacteria to cure them or prevent infection with white nose syndrome. But that's a really logistically difficult thing to do. So I think they've struggled with that. But in the lab, at least, yes, there are 2 bacterial genera that have antifungal properties that came from the endogenous skin microbiome of wild bats. Yeah.
Cool. Thank you.
Okay, we have another question in the chat from Elise. Thanks, Melissa, for this amazing presentation. Between bat feeding guilds, did you find clear microbiome differences?
Yeah, so it's kind of an interesting question. At the taxonomic level, less so. So bats are really strange compared to other mammals in a variety of ways, and their microbiomes are totally consistent in that trend. So bats don't have clear associations with specific microbes to the extent that other mammals do. But one thing that was kind of surprising is that while taxonomically, it's not that fruit eaters have more of this species than nectar feeders and so on. At the functional level, we definitely see those things. So I think this is another byproduct of that functional redundancy in microbes, where bats that are frugivorous can have very different suites of bacterial species that are all accomplishing the same thing. And those functions are very different between the guilds. So fruit-eating bats have a higher enrichment of essential amino acid pathways in their gut microbiomes, which makes sense because fruits are usually pretty protein deficient food resources, right? We don't see that same enrichment in insect-eating bats, for example, because their diet is mostly protein. They don't need that supplementation as much. So it's this very interesting divide between signal we can detect with the bacterial taxonomy and signals we can detect with the functions.
Great question. Thanks, Elise. Anybody else? Anybody else? Well, I welcome anybody to also contact Melissa later on for any future questions, if there are any. I would also like to mention that in March, we will have our second webinar. So final round of applause first for Melissa. Thank you again for doing this. Really appreciate you being our first guinea pig for this. And next month, we will be talking about disease ecology, and that will be on March 29th at the same time. So that's also a Wednesday at the same time slot. I will send out a flyer and you will be able to RSVP again in the same way. Our speaker will be Gwen Kettenberg, who is in the crowd. She's one of our GBATnet students. And yeah, again, this will be on the YouTube afterwards. So if you would like to find, yourself asking yourself some questions and going back to it, please check the YouTube again. Melissa will share some of the slides so that you can also refer to them. And if you have any other questions, we will put those also as resources so that you can ask Melissa, and she'll provide some sort of answers that you can really get at the research and hopefully participate in this in the near future. So I'm going to stop recording right now. And